Slides.
Good morning, everyone. Thank you to the organizers for giving me this opportunity to give a talk here. I'm Angana Mukherjee, and I come from Laval University, Quebec City, and I work with Dr. Dave Richard. So I'm going to talk about systemic functional analysis of the phosphoinositide metabolic pathway in Plasmodium falciparum. So what are phosphoinositides or PIPs? These are negatively charged glycerophospholipids present in the cytosolic surface of the membranes or their phosphorylated forms of phosphatidylinositol. Although they account for less than 1% of cellular lipids, they are critical components of cellular membrane. They play important roles in various kinds of processes from signal transduction, cell motility, cytoskeletal organization, DNA synthesis, membrane transport, permeability, and trafficking. And depending on the position of the phosphorylated group on the myoinositol head group, they are identified as 7 different species. So they can be monophosphates like PI3P, PI4P, PI5P. They can be diphosphates like PI34P2, 35P2, 45P2, or triphosphate such as PI345P3. Now, these 7 forms of PIPs are generated or modified by PIP kinases or PIP phosphatases, which are named based on the position of the substrate they are phosphorylating or dephosphorylating. And here, these are some examples of PIP effectors. They have lipid binding domains such as pH or pX domains, and they bind with differential selectivity and affinity to various PIPs on cell membranes. So in eukaryotes, I think as much as 15 different PIP binding domains have been described. And because they bind to PIPs, and then they also bind to cellular signaling molecules. And so this collectively, these 7 isoforms of PIPs, the kinases and the phosphatase, and these PIP recognizing effectors, they form a remarkable complex protein lipid signaling network. So that's about eukaryotes. What do we do about PIP profile, localization, and functions in Plasmodium falciparum? A lab has recently localized several species of PIPs in the asexual stages of Plasmodium falciparum. We, with others, have shown that PI3P is on the food vacuole membrane and also in epicoplast. Others have shown the importance of PI3P in hemoglobin uptake to the food vacuole, biogenesis of epicoplast, and it has also come up to be associated with artemisinin resistance. We have shown that PI4P is present on the plasma membrane and in Golgi apparatus, and Elizabeth Winsler's group has shown that it's PI4 kinase and PI4P is important for proper plasma membrane invasion during schizogony. They've shown that imidapyrazines affect PI4P distribution, and it has also been associated with artemisinin resistance, a paper that has come up from Dan Nifsy's lab. PI45P2, we have shown it is present on the plasma membrane and also in cavity-like structures, and then it's responsible for calcium signaling cascades for gametocyte activation, merozoite egress, and invasion. So with this in mind, about a year ago, we decided to perform a comprehensive knockout screen wherein we attempted to disrupt the complement of the PIP metabolic pathway, including 8 PIP kinases, 3 phosphatases, about 14 PIP effectors, and 3 PIP transfer proteins. So these are 29 targets. We wanted to identify the genes that are essential for asexual growth in Plasmodium falciparum. Okay. All right, for this, we utilized a simple and robust technique that has been recently reported in Nature Methods by the Spielman group. So it's a paper on Nature Methods. It came out in 2017. So they call this targeted gene disruption by selection-linked integration. permitting selection of parasites with genomic integration. So in this approach, essentially, a promoter-less part of the target region on the plasmid that has to be integrated is separated by an additional antibiotic resistance marker, in our case, neomycin phosphotransferase. And neomycin phosphotransferase will only express when it has recombined into its homologous recombination by a single crossover. So the target is linked to it, but is separated by a skip peptide. All right, and so first, so we selected for, so after transfection, we selected for parasites carrying episomes with WR, and then we selected for the integrants with NEO. Okay, and so we had 4 different kind of consequences. First, we could not recover integrants after selecting for NEO. Oh, and then to remind you, we carried out 3 independent attempts to target each gene. All right, so if we didn't recover any parasites after neo-selection, we concluded that it's probably essential. And then if we recovered parasites, we monitored correct integration and loss of original locus by doing PCR at the 5' junction, 3' junction, and looking for loss of wild-type copy. So we had 3 examples. Example 1, where we recovered parasites, but we didn't see any integration at the 5' or 3' junction, and we saw the presence of the original locus in whichever selections we recovered. So we conclude that it's probably a likely it's probably an essential gene. In our second example, we do see 5' and 3' integration, but we also see the presence of the original locus, and so probably the locus has duplicated, and so probably it's also an essential gene. And in our third example, we see correct integration in all our, so these are different selections. So we see correct integration in our knockouts, 5' and 3' integration, and we see loss of the original locus, and so that's an example of a dispensable gene. All right, and so now coming back to the same list. So our study showed that 72% of the genes of the PIP metabolic pathway could not be disrupted and are likely to be essential for the maintenance of parasite viability, and that correlated well with the sparse essentiality data we have from homologs in P. Berghii or Toxo. In black are the genes that we could not disrupt, so they're essential. In red are the genes that we could get knockouts of, and green boxes are the ones that agreed with literature, whatever literature we had from P. Berghii or Toxo. So if it's a white box, doesn't mean that we have controversial data. It's mostly because there is no data about that gene. All right. And so this lack of redundancy points not only to the vital role of the number of individual kinases, effectors, phosphatases, but also a potentially, abundant source of anti-malarial targets. Okay, so now I will show you data of asexual growth in each of these knockouts. All right, first PIP transfer proteins. On top you see PCR images showing correct 5' and 3' integration and loss of original locus, and at the bottom you see parasitemia determined by flow cytometry over 3 cycles. So out of the 3 PIP transfer proteins, we noticed a growth defect in this knockout of a PIP transfer protein, during asexual growth. Coming to knockouts of PIP kinase, phosphatase, and effectors, there wasn't so again we confirmed correct integration and a loss of wild-type copy, and there wasn't a difference in the growth of any of these knockouts compared to the parental line. This is one PIP effector where there was a slight difference in growth in the knockout later in the 3rd cycle. Okay, so next coming to knockout of this PIP binding protein, 72700. It's a PIP binding protein with a PX domain and 3 transmembrane domains there. First, we confirmed again correct integration at the 5' and 3' junctions in all the 3 independent knockout selections. We confirmed loss of wild type locus in all the 3 independent knockout selections. And then to determine the requirement for this PIP binding protein during asexual parasite development, we monitored replication of 2 independent knockouts by flow cytometry over 2 cycles. Knockouts demonstrated a 3-fold decrease in parasitemia compared to the parental line. Now this reduced growth rate could be due to reduced viability, could be due to reduced invasion rate, could be due to less merozoite formation by segmentation, and it could be a smaller progeny of schizons. So to gain insight into the basis for attenuated growth, we monitored the appearance of developmental stages during asexual proliferation. So what did we do? We purified mature schizons. We incubated with fresh RBCs for 5 hours. So then 0 to 5 hour rings were sorbitol synchronized. Then we plated equal number of rings, and the development was followed by light microscopy till 61 hours. So the knockouts did not seem to develop at the same rate like the parental line. So when you see trophozoites in the wild type, you still see a subset that is delayed and are rings. When they are schizons, you still see them as trophozoites, and when 3D7 has formed mid to late rings, they're still at schizons and some early links. And a major part of the population appeared abnormal and formed pycnotics. So we quantified the stages, and we expressed them as percentage of stages. And so you again see that 40% of the population in the knockout appeared abnormal and pycnotic, and you can now quantify the delay. So here, when they are trophozoites in 3D7, a subset is still at rings, a small subset has formed trophozoite, and then when mostly at 40 to 45 hours, when 3D7 is schizon, you see them as trophozoite and late rings, and then when these are mid or late rings, they finally matured, the knockout finally matured into schizons and early rings. And you also see like the 40% look abnormal or pycnotic. All right? And so we thought it was important to localize this protein. So we again used almost the same technique, selection-linked integration, where we C-terminally, we tagged the PIP binding protein C-terminally with GFP. And so in later stages of asexual development in trophozoite and in schizon, we saw a part of GFP fluorescence surrounding the food vacuole. And so we concluded that this PIP binding protein, at least partially, is localized to the food vacuole membrane in later stages. And this made sense because PX domain that is renowned to bind to PI3P is present in this protein, and we and others have mapped PI3P to the food vacuole membrane. So it's not surprising that PIP binding protein is localizing partially to the food vacuole membrane. Okay, then sorry, because the role of PI4K and PI3K forming PI4P and PI3P respectively in artemisinin resistance, we thought it made sense to evaluate the susceptibility of these knockouts to antimalarials. So we took 2 independent knockouts of PIP binding We took 2 independent knockouts of PIP binding protein, and we also included the PIP kinase knockout that we got. 1 minute. Yeah. And then evaluated their sensitivity to antimalarials. We found a little bit modest increased sensitivity to lumefantrine, mefloquine, piperacin, and DHA, but not chloroquine and amodiaquine. I'll just touch upon knock sideways. It's another approach that we have used for functional analysis of proteins, but essential proteins. And in this case, it's a PIP effector with a pH domain protein. So how does that work? It's a ligand-induced dimerization of the proteins FRB and FKBP. The native target is fused to FKBP, and the FRB is expressed separately with the trafficking information in a different cellular compartment, the mislocalizer. When you add a ligand such as rapamycin will induce dimerization of FKBP, and FRB, and will remove the protein from its site of action into another compartment. And therefore, that's how you can functionally analyze essential gene. And so just one more slide and I'm done. And so we used it to functionally analyze an essential PIP effector with a pH binding domain. And so as you can appreciate, in the presence of FKBP and FRB that are expressed separately on plasmids, you can see that the GFP fluorescence is very different from the mCherry or DAPI, suggesting that this protein is expressed elsewhere. But then when you add rapamycin, the GFP fluorescence pattern changes and is kind of translocated to the nucleus. So we know knock sideways has happened. It has moved the protein from its site of action, and the parasites did not appear healthy. And so when we did a growth curve, we saw in the presence of rapamycin, they have decreased parasitemia compared to all the controls. All right, and so I'll just quickly summarize.
We're done.
We are done. We are done. So that's the summary. There's a long way to go. We just started this project a year ago. We are currently functionally analyzing all the essential proteins. All right, with that, I'd like to acknowledge everyone who has contributed.
Sorry, we have finished. No. Thank you.